Welcome to Quicksand, the most important news in pharma and biotech this week. The FDA released draft guidance to help sponsors design and conduct first-in-human clinical trials that speed the clinical development of cancer drugs through multiple expansion cohort study designs. The guidance provides FDA's thoughts on the characteristics of drugs and biologics appropriately suited for such an expedited plan, information to include in such investigational new drug application submissions, when to interact with FDA, and safeguards to protect patients enrolled in first-in-human expansion cohort studies. First-in-human multiple expansion cohort trials, defined as trials with a single protocol with an initial dose escalation phase that also contains 3 or more additional patient cohorts with cohort-specific objectives, are intended to expedite development by proceeding from initial determination of a potentially effective dose to individual cohorts that have trial objectives typical of phase 2 trials, FDA says. The objectives of these expansion cohorts can include assessment of anti-tumor activity in a disease-specific setting, assessment of a reasonably safe dose in specific populations, evaluation of alternative doses or schedules, establishment of dose and schedule for the investigational drug administered with another oncology drug, or evaluation of the predictive value of a potential biomarker, the draft says. First-in-human multiple expansion cohort studies pose unique challenges and risks, according to FDA, including exposing patients across multiple simultaneously accruing cohorts to potentially suboptimal or toxic doses of an investigational drug, or exposing more patients than required to achieve certain objectives. To ensure such studies are worth the added risk, FDA says the patient population should be limited to patients with serious diseases for which no curative therapies are available. The 14-page draft guidance also features sections on considerations based on cohort objectives, statistical and safety considerations, as well as protocol content and communications in interactions with FDA. The FDA released new draft guidance outlining the clinical endpoints acceptable to demonstrate effectiveness for medication-assisted treatment of opioid use disorder. The 8-page guidance, following draft guidance from April on the development and clinical trial design issues relevant to the study of sustained-release depot buprenorphine products, discusses clinical endpoints including adverse outcomes of opioid use disorder, change in disease status using diagnostic criteria for opioid use disorder, patient-reported outcomes, change in drug use pattern, and other outcome measures. FDA encourages sponsors to consider a variety of ways to evaluate the effect and clinical benefit of medication-assisted treatment, including impact on mortality, emergency medical interventions, and hepatitis C seroconversion. Efficacy may also be measured by studying the proportion of patients that transition from meeting criteria for being diagnosed with moderate to severe opioid use disorder, based on both drug use and its impact on patient well-being at baseline to being considered in remission at the end of study. Improvements in the ability to resume work, school, or other productive activity may also demonstrate clinical benefit, the agency added. In addition to the draft, FDA also recently published a paper with the National Institute on Drug Abuse that describes efforts to overcome some of the barriers to new drug development and the issues with determining effectiveness. The FDA released final guidance for sponsors on when a standard release test and criteria may be used in lieu of extensive method development and acceptance criteria setting exercises. The 8-page guidance, FDA said, establishes standard dissolution methodology and acceptance criteria that are appropriate for highly soluble drug substances that are formulated in immediate release dosage form. The availability of these standards will facilitate the rapid development of dissolution methodology and related acceptance criteria, with no requirement to show discriminatory ability of the dissolution method for these products during drug product development. In addition, these standards will facilitate FDA's evaluation of the data submitted in the application, FDA added. In general, knowledge about the solubility, permeability, dissolution, and pharmacokinetics of a drug product is considered when defining dissolution acceptance criteria for the drug approval process, FDA explained. For drug substances that do not meet the condition in this guidance, FDA said that sponsors or applicants should follow the recommendations provided in the August 1997 guidance, dissolution testing of immediate release solid oral dosage forms. The FDA announced it plans to hold a public discussion on expanding the National Drug Code format and the impact on industry. FDA currently assigns 10-digit NDCs for the purposes of identifying drugs in the country, and the format is comprised of 3 codes regarding the labeler, the product, and the package. FDA anticipates that it will run out of 5-digit labeler codes in approximately 15 years, the agency wrote in the notice on the November 5th public hearing. FDA will begin assigning 6-digit labeler codes at some point in the future due to exhaustion of 5-digit labeler codes. Prior to implementing the new formats for longer configurations, FDA officials will seek feedback from meeting participants on the potential impact on companies' bottom line and drug safety, as well as issues associated with the current lack of NDC uniformity in the marketplace. The agency said the purpose of the hearing is also centered on discussing requests it has previously received from stakeholders to adopt a standardized NDC format. In 2013, FDA rejected a petition to stop using NDCs for unapproved drug products, thwarting the concerns by arguing the codes make it easier to identify drugs regardless of their approval status. The European Commission opened for consultation a new draft guideline on good clinical practices for advanced therapy medicinal products, or ATMPs. The new 14-page draft guideline, which created in consultation with the European Medicines Agency and is specific to clinical trials that are conducted with ATMPs, which may be complex to design and conduct. For example, manufacturing constraints and the short shelf life of the product may require the implementation of tight controls on logistical arrangements to administer the product, the draft says. Likewise, the mode of application may render very difficult the use of placebo controls and/or require specific training. Additionally, the long-term effects of the product may require specific arrangements for long-term follow-up of the subjects. Moreover, it is recognized that it may not always be feasible to generate relevant preclinical data before the product is tested in humans. GCPs may therefore need to be adapted to specific characteristics of ATMPs, the guideline says. The guideline features sections on clinical trial design, application dossier, specific considerations concerning the protocol in investigator's brochure, quality of the investigational ATMP, administration procedures, traceability, retention of samples, protection of clinical trial subjects, safety reporting, and monitoring. Thank you for listening to Quicksand, episode 42, August 13th, 2018.